morning.
Good morning.
Yes, Peter.
Good morning.
Yeah.
Peter.
Good morning, sir.
Good morning.
Good morning,
Yes.
sir.
Yes, Dr. Susan is my classmate
Oh,
at Maharaja's College, Ernakulam.
Peter, sir.
Yes, yes, yes.
How are you? Nice to see you.
I am fine, very fine. Very
Yeah,
fine.
last time we met in NAS, National
Yes.
Academy Agricultural
Yes,
Sciences,
yes, yes.
in Delhi. Yeah.
Yes, yes, yes. The world is so small, you know.
Hello.
This internet has made a world smaller.
Yeah. That's true. That's true. Yeah.
Actually, what happened today, it started
Okay.
3 days back, an
Uh-huh.
international congress on horticulture in TNAU.
Oh, I see. Okay.
Coimbatore.
Okay.
So I had
Uh-huh.
to chair a session at 10 o'clock. So
Oh.
I requested Dr. Susan to
Uh-huh.
give me 1 minute only to introduce about NASI. We have
Okay.
here National Vijayan sir is a honorable fellow of NASI,
Uh-huh.
and he is the whole coordinator.
Okay.
So when he is there, I cannot go away to Coimbatore. So I told Susan, in any case, I have to reach there at 10 o'clock.
Okay.
So I will speak just the introduction of NASI. What is
Okay.
National Academy of Science India?
Okay.
And about Dr. Saha, 2 minutes.
Okay.
And I will. How are you, sir?
Yeah.
How are
Fine.
you?
Fine. Yeah.
Fine.
Hello. Yeah. One
Yes,
minute.
sir.
Yeah.
I'm
I'm
hearing you.
fine, and I'm in Philadelphia.
Okay. Okay.
So well, I came here about six weeks back. Yeah.
Okay.
I'm here. Yeah.
Oh,
I'm here.
very nice.
My family is here, so
Oh, very
I
nice.
visit them.
Philadelphia is the center of knowledge and science communication.
Oh, yeah.
I was in 1982. I was with NAL, National Agriculture Library, Bellsville.
Okay.
NAL Bellsville, National
Okay.
Agriculture Library, Bellsville. So at that
Okay.
time, this Philadelphia,
Yeah.
all materials they get from Philadelphia. Of course,
Yeah.
computer has started at that time, but not like
Mhm.
this.
Mhm.
Very good.
One more one.
Shruti, ma'am, can we start? Yeah, just wait for 30 seconds. Okay. People are joining. Yeah, let them to. Correct. Ashwati, you can start.
Okay, ma'am, am I audible?
Yes.
Yes, you are.
In learning, you will teach, and in teaching, you will learn. Is a quote by Phil Collins that encapsulates within it the beauty and art of effective teaching. Greetings of the day and a hearty welcome to everyone present here. I deem it a great joy to have all of us gathered here. Hope you all are good and safe. Thank you for taking your valuable time out and being here with us from all around the world. A gentle reminder, please follow the instructions during the webinar for the smooth functioning of the program. Be sure to mute your audio so that there is no disturbance during the sessions. If you have any queries, please type those into the Zoom or YouTube live chat box. You may send in your questions at any time during the presentation. We will collect these and address them during the Q&A session at the end of today's presentation. The feedback form of the webinar will be available at the end of program. Department of Biosciences, Union Christian College, Aluva, Kerala, and National Academy of Sciences, India, proudly presents to a webinar on the topic Metabolic Engineering of Plant Secondary Metabolites for Value Addition with reference to economically important crops and champion algal forms by Dr. G.A. Ravishankar, the Vice President and Professor of Biotechnology, Dayananda Sagar Institutions, Bengaluru, as a part of our ongoing Centenary webinar lecture series. We have our beloved Principal Dr. Tara K. Zaiban, Manager Reverend Father Thomas John, former BARC scientist and adjunct faculty member Dr. Susan Epen, Head of Department of Biosciences Mr. Shyam Mohan, Faculty Coordinator of the program Dr. Serene Sarah John, and other faculty members of UC College. Before we begin, let us invoke the Almighty's presence and blessings in this gathering, for which I request you all to close your eyes for silent prayer. Now, I invite Dr. Susan Epen to introduce our respected dignitaries for the welcome address.
A very good morning to all of you. It gives me immense pleasure to welcome you all to this webinar organized by National Academy of Sciences, India, Prayagraj, Kerala chapter, and hosted by Union Christian College, Aluva, Kerala, which is completing 100 years since its inception. First of all, let me welcome today's distinguished speaker, Dr. G.A. Ravishankar, to this program. Welcome, Dr. Ravishankar. Former Vice Chancellor, Kerala Agriculture University, and former Director, ICAR, IISR Calicut, and Chairman, NASI Kerala Chapter. Ladies and gentlemen, we are privileged to have Professor K.V. Peter with us. Welcome, sir, to this August gathering. I also use this opportunity to welcome Professor Edathil Vijayan, former ICMR medical scientist and UGC visiting professor and convener of today's program. Welcome, Dr. Vijayan. I'm extremely pleased to welcome Dr. Deepu Matthew, Secretary, NASI Kerala Chapter, and all other distinguished NASI fellows and members to this meeting. I also welcome UC College Manager, Reverend Thomas John, and Principal Dr. Tara K. Simon, and all the faculty from Union Christian College. Finally, I welcome all the participants from not only India, but also from abroad for today's webinar. Thank you all.
Thank you, ma'am. Now, I would like to invite Professor K.V. Peter, President of NASI, for a talk.
Sir, it seems Peter sir has some problem. He told he is traveling. In his absence, can I talk about NASI?
Yes, yes, sir.
Yeah.
It's okay.
Thank you so much. Respected Ravishankar sir, Susan madam, Vijayan sir, distinguished fellows and members of NASI, and the faculty of UC College. A warm good morning. At the outset, let me congratulate Susan madam and the entire faculty of the Department of Bioscience for organizing such a beautiful webinar. A national
Sarin, can you call Deepu?
Yes. Deepu, sir, I think his network connection is gone now. Yes, I'll call.
Ashwati, can you ask Dr. Vijayan to proceed?
Yeah.
Yes.
Am I audible, madam?
Deepu, who is
Yes,
that?
you are.
Yes, you are.
Deepu has come back. He
Fine,
can
yeah.
proceed. Yeah.
So there was some technical snag. Sorry for that. Yeah, right from its inception, the National Academy of Sciences has been continuously striving to serve to this objective. And as of now, throughout the country, we have different chapters. And the Kerala chapter was established, and maybe this is the youngest chapter for NASI. And the Kerala chapter was established in 2019. And we are so happy to get associated with the prestigious colleges as well as organizations. And we are all out to sponsor as well as support every kind of scientific activity which can serve our society. Very happy to see that an eminent scientist, Professor G.A. Ravishankar, is the major speaker today. And he is a well-known scientist in plant, algal, and food biotechnology, as well as in the area of plant secondary metabolites. So wishing every success for the program and congratulating the organizers once again. Thank you so much.
Thank you, sir.
Ashwati, you are on mute.
Sorry, ma'am. Thank you, sir. To give us the opening remarks for this program, may I now call on Dr. Genetically Vijayan, NASI.
Very good morning to all. And Professor Peter, Principal and Management of UC College, Dr. Susan, and faculty, students of Bioscience Department of UC College. It's a great privilege to address you this morning, and I congratulate UC College and especially Dr. Susan Epan for taking this initiative and getting the opportunity of Kerala chapter of National Academy of Science to organize this wonderful lecture by one of my close friend and colleague, Dr. Ravishankar. And the Academy, as Dr. Deepu said, the Kerala chapter of the National Academy of Science is the youngest chapter. We have about 18 or 19 chapters across the country representing the National Academy of Science. And primarily, the NASI, we call it National Academy of Science. NASI is mandated to cultivate, promote science and technology through all branches for promotion of social welfare and scientific temper in the country. Taking science to the unreached and promotion of science to schools and colleges by supporting lectures by eminent scientists, organizing workshops, demonstrations, and scientific exhibitions in rural and backward areas. And today's lecture by Dr. Ravishankar, who is an outstanding plant molecular biologist, is one in that direction. And I'm sure this will be, Kerala chapter has been supporting activities like this in many institutions, and by word of mouth and now by communication, the awareness of the existence of Kerala chapter of NASI and the role played by the Kerala chapter in promoting science and technology in the especially in Kerala is catching up, and it is known. So we get now more and more requests, inquiries in order to conduct such activities. It's an excellent sign, and probably UC College Aluva during its centenary celebration, and I'm sure, I think this is our second program which we are supporting in UC College. So the NASI Kerala chapter is very proud to be associated with the UC College of 100 years old, and in whatever little way the National Academy of Science Kerala chapter will be supporting such activities in other institutions as well. I utilize this opportunity to, on behalf of the Academy, on behalf of Kerala chapter, to thank everyone of UC College. And I also thank Professor Dr. Ravishankar in agreeing to deliver this lecture. With these few words, I wish all the success and hope the students and faculty encourage such activities in other institutions as well. Thank you.
Thank you, sir. Thank you, sir. I would now call upon Dr. Susan Epen, former Bach scientist and our adjunct faculty, to introduce our guest speaker for the day.
International consultant in World Bank Project USA, Vice President of R&D in Life Sciences and Biotechnology, Dayanand Sagar Institutions, Bengaluru, former Director Grade Scientist and Head of Plant Cell Biotechnology, CFTRI, Mysuru, international expert in areas of plant biotechnology, algal biotechnology, food biotechnology, post-harvest technology, plant secondary metabolites, functional foods, herbal products, and genetic engineering, a visiting professor to different universities in Japan, Korea, Taiwan, and Russia, a guide who has guided 27 PhD students, a person with 260 peer-reviewed papers, review papers 48, who has editor of 5 books, with a research H index of 68 with the citations of 20,000. The person who has delivered 250 lectures with 55 patents in India and abroad, a fellow of several academies like fellow of National Academy of Sciences, National Academy of Agricultural Sciences, Association of Microbiologists of India, Society of Agricultural Biochemists, Society for Applied Biotechnology, Indian Botanical Society, Biotechnology Research Societies of India, etc., etc. The person who received different fellowships from different agencies like International Academy of Food Science and Technology, Canada, Institute of Food Technologies, USA, Institute of Food Science and Technology, UK, and Certified Food Scientist of USA. A person who has received so many awards like Professor Hira Lal Chakrabarti Award, National Technology Day Award of Department of Biotechnology, Lalji Gudu Gudbu Samarkar Nidhi Award, Subramaniam Industrial Achievement Award, Professor Vyas Memorial Award, SS Katiyar Memorial Award, Lifetime Achievement Award, Professor Gadgil Memorial Lecture Award, and also featured last month in the Biotechnology Express as the Biotech Heroes of India in the cover page. Ladies and gentlemen, we are today going to listen to Professor G.A. Ravishankar, who will enlighten us on metabolic engineering of plant secondary metabolites for value addition with reference to economically important crops and champion algal forms. Dr. Ravishankar, please.
Sir, you are on mute, sir.
Thank you, Dr. Susan, for a very elaborate introduction. Respected principal, invitees, participants, faculty, students of various institutions, including the prestigious UC College, Dr. Peter, Dr. Vijayan sir, and the organizers of this webinar, and the participants from all over the globe, as I was told, and also students of Bioscience Department, ladies and gentlemen. At the outset, I wish to thank the management of Union Christian College for inviting me to this webinar in connection with the Centenary year, and also in this webinar, which is in association with the National Academy of Sciences. And I've been associated with the National Academy of Sciences for several decades. So I'm thankful to the Academy for this support. And I'm also thankful to many colleagues, Deepu Matthews, Dr. Sareen, Dr. Ashwati, and many others who are involved in this function and in this webinar of today. So with this, I wish to start my presentation. Is it seen?
Yes.
Okay.
Yeah. Yeah.
Is it okay?
Yes, yes, it's perfect.
Okay. Yeah, I chose this topic, metabolic engineering of plant secondary metabolites, and with special reference to the croplands and algal forms for two reasons. One is that I worked on secondary metabolites ever since my PhD time in Baroda. And then I continued with it in Jammu when I joined CSIR lab there. And again, after joining CFTRI, I started working on the secondary metabolites of food value. And I also joined a team working with the algal forms, especially the microalgal forms. And we developed technologies related to the higher plants for secondary metabolites of economic value, and also algae of economically important forms which are used in food and medicines and so on. So therefore, I thought this topic will be of broader interest to the students and to the audience in general. And so what I would like to do today is that I give a background on the phytochemicals and its use as drugs, and then how plant tissue and cell cultures could be used for the production of secondary metabolites. And then what are the strategies involved in enhancing the secondary metabolite production under in vitro conditions, and how to increase the secondary metabolites by challenging them with some of the biotic elicitors. I'll explain to you the concept of all these. And then I come to, this forms the background for my further presentation on the metabolic engineering of secondary metabolite pathway. I have some stories from my own research and research involving a lot of PhD students, a number of my colleagues. So I owe a lot to all of them when I make this presentation, which in fact is distilled from more than 20 PhD theses which has come out from our group. And therefore, thanking all of them, I would like to make this presentation. And this is how I have structured my presentation for today. Coming to the secondary metabolites, those of you who are not familiar with this, I would like to say that these are the metabolites which are synthesized from the basic photosynthetes. And the elaboration of the photosynthete into complex molecules called secondary metabolites, they are of use in food, medicine, healthcare products, chemicals of multiple uses. And it is said that these secondary metabolites are not essential for growth and life of the plant, but rather required for interaction of plants with its environment and produced as a result of the response to the environmental stresses. It could be biotic stress or it could be abiotic stress, which will influence the formation of these metabolites. So from the basic photosynthete, there is branching off to various complex pathways, and you will have the formation of alkaloids, you will have the formation of phenolics, you will have the formation of terpenoids, you have the formation of sterols, and several such complex molecules. And you know that many of these molecules are bioactive molecules and are of use for various functional properties. And these metabolites are also influenced by abiotic stresses, because the plant is exposed to the abiotic stresses and the biotic stresses. And this also influences the metabolite production. And it's when we are trying to regulate this metabolite production in the plants, we need to look at these abiotic signals and also the biotic signals. So this, we summarized the abiotic signal pathways in this particular publication, which came in 2011. It's one of the highly cited papers. It has received nearly 2,000 citations so far. These secondary metabolites are present in abundance in almost all the plants, and the plants have been used as herbal medicines in Ayurvedic system of medicine, or the Chinese system of medicine, or in medicines in various other parts of the world, maybe in South America and so on. And it has been in usage for centuries. In spite of the advancement in the synthetic chemistry, we depend on the plants for various drugs. For example, artemisinin from Artemisia for malaria treatment, capsaicin for pain management, colchicine for gout, cannabinol for chronic neuropathies, and so on and so forth. So therefore, these are some of the indispensable molecules which are obtained from plant sources, which have been used in various for various medicinal properties. And also, there are many plant-derived metabolites which are in advanced clinical trials. For example, beta-carotene for tumors, curcumin for anti-cancer, inflammation, and for anti-inflammatory activity, and for Alzheimer's and so on. Epigalactogen, 14 trials on cancer and Alzheimer's. So Genetically, 5 trials, Gossypol, 2 trials, quercetin, 9 trials going on, resveratrol, which is a nutraceutical, which is from Vitis vinifera, more than 22 trials happening worldwide to establish the efficacy and also the toxicity and so on and so forth, so that it can be used as drug or it can be used as nutraceutical and so on. So some of the outstanding examples of indispensable plant-derived natural products, you have the examples of Taxol from Texas, Becata, vincristine, vinblastine, camptothecin, artemisinin again. So these are the molecules which are presently used in therapies and which are derived from plants. And therefore, it's important to know that these drugs are indispensable, and they can be, and they have to be obtained from the plants. And there are now novel methods to obtain this using the modern tools of in vitro culture technology, or metabolic engineering, and so on, which I'm going to describe with few examples. In fact, plant cell tissue and organ culture will provide an alternative to production of these metabolites because the plant cell is chemically totipotent. That means it can produce the whole range of compounds which is produced in the intact plant. And today, with the understanding of the metabolite pathway and also the genomics of the metabolite production pathway, and especially the functional genomics with specificity of the each step, we are now able to even manipulate this in a desired manner. And that's the main aspect of the pathway engineering. And it provides a number of opportunities to regulate the gene, and also through a synthetic biology approach, you can produce novel compounds and molecules in plant tissue and cell cultures. And also you can also produce them in heterologous hosts. So it could be in bacteria, yeast, and so on. The production of these secondary metabolites can be done through callus cultures or cell suspension cultures. You can also immobilize the cells which produce these metabolites and then grow them on a continuous mode. You can also use this for biotransformation, that is conversion of a lesser value compound to higher value compound. And this has been elegantly demonstrated in case of digitoxin and digoxin, which are, digoxin is a molecule for cardiac diseases. So it has been already commercialized. And then hairy root cultures, that is root-derived compounds, can be obtained through Agrobacterium rhizogene mediated transformation system. And elicitation, when you challenge the cells with biotic elicitors or abiotic elicitors, it produces it induces the formation of the secondary metabolites in several folds higher than the baseline. And you can also make these cells to permeate the produced compound, and you can grow them in bioreactors. And today, with the power of metabolic engineering, you can engineer the pathway, you can in the desired manner, and get high amount of the secondary metabolite which is required, or value add to the plant through metabolic engineering. And then the synthetic biology approach is the latest trend, where you can borrow the gene from various different sources and then put them in a cassette, which will be able to produce the metabolites in very high levels, so that it can be commercially feasible to produce them through biotechnology. And then delivery of bioactives and in silico analysis. And some of these we have presented in this review, Plant Cell Culture as Chemical Factories. It's again one of the highly cited papers from our group with over 2,000 citations as of today. And the group of secondary metabolites could be phenolic, phenyl propanide compounds, alkaloids, terpenoids, quinones, steroids, and so on. So I've listed here various compounds which are being which are produced in the in vitro under using plant cell and tissue cultures. In fact, we our group has worked on a number of metabolites. We worked on anthocyanin production, betaline production, and we developed processes for production of these in bioreactors. And so also many other groups have worked on cell cultures. And these are the examples which are commercially produced cell cultures, which produce chiconin, in this case, from Lithospermum erythrorhizon, and afterquinone, which is used for some bacterial infections. And also it is used as since it's a dye, it's also used as used in cosmetics and so on and so forth. And this has been commercialized. And so therefore, the cell cultures can be produced and can be made to produce high quantities of metabolites, and they can also be grown in bioreactors. And very many techniques which I mentioned, which I listed here, will be useful in enhancing the production of these secondary metabolites. In fact, our work has been well recognized in terms of with respect to some awards. And this is the one now which I received from the Prime Minister when this is based on the studies which I had done on plant tissue and cell cultures for secondary metabolite production. These secondary metabolites can also be produced through hairy roots. Hairy roots are obtained by infecting the plant with the Agrobacterium rhizogenes. If you infect the plant with the Agrobacterium rhizogene, you have the formation of the hairy roots. That's because of the root inducing trait of the Rnai plasmid, which is in the Agrobacterium. And here you have an example of Withania somnifera with a prolific formation of the hairy roots here. We have grown this in the bioreactor. And also, this produces Vitaferin A. Vitaferin A is an anti-tumor compound. And not only that, you can produce in high quantities in a continuous mode. If you challenge these cells, these hairy roots with elicitors, here salicylic acid, when you challenge with, you will have very high production, 40-fold increase, 42-fold increase in the production of the Vitaferin. So these are some good examples to show that they are very promising techniques and can be adopted to develop technologies for high-value metabolites. And these hairy root cultures have been commercialized already for ginseng. And as you know, the ginseng is a very slow-growing plant, and it has many medicinal uses. And this can be produced in through hairy root cultures in bioreactors, and it has been scaled up in Japan and Korea. So essentially, the cell cultures or the hairy root cultures can be scaled up, and you can produce these natural compounds. Some of the ones which are commercially produced today are the Taxol, ginseng, anthocyanins, and so on. And the factors which influence the production are the elicitation, permeabilization, immobilization, biotransformation. These are some of the techniques which can be used for enhancing the production. And this is a very crowded slide. I want to tell you that the plant tissue and cell culture have been scaled up, and the cell culture derived food, pharmaceutical or cosmeceutical molecules have been produced by various commercial companies in Japan, in Germany, in Switzerland, and US, and so on. So this is to tell you that it is now produced in commercial scale in bioreactors, and the compounds are extracted and are used for various intended uses. So now I come to another aspect of enhancement of the metabolite in cell cultures, and that is through elicitation. That means in this method, the cells are challenged with some of the fungal or bacterial derived molecules, or even the bacterial or the fungal extract itself can be used as elicitors. If you challenge the cells with these elicitors, the metabolite yield will be higher. And this is, as you know, the plants, when they are attacked by any of the microbes, they produce phytoalexins in response to the infection. And a similar thing happens with when you challenge the plant cell with elicitors. It need not be elicitor, need not be from a pathogenic source. It could be a non-pathogenic as well. But the fungal polysaccharides and some of the components in the cell, in the fungi or bacteria, influences the formation of the secondary metabolites in very high quantities. Even abiotic elicitors like jasmonic acid, salicylic acid, ascorbic acid, when they are challenged, you have the spike in the production of secondary metabolites. So you have examples of various plant cell cultures responding to different types of felicitors to produce a range of products like phytoalexins, sesquiterpenoids. You have diasgenin, ginsenosides, Taxol, azadirachtin, and so on from various examples. I will now tell you a story how we use this elicitation for increasing one of the very important metabolites, that is bixin and norbixin. These are the carotenoids produced in plant called Bixa orellana. Bixa orellana is a plant of South American origin, and in India, the cultivation of this plant is undertaken in Andhra Pradesh and in some parts of Karnataka. And what we did was we found using cell cultures model, we found that Aspergillus niger and Rhizopus oligosporus extracts would enhance the formation of bixin. So we have published this study, and after that, what we did was we used this elicitor to challenge the plant, the standing crop of Bixa orellana, and we could see that there is increase in the yield of bixin by almost 3 to 4 folds. See, originally the Indian Bixa plants were producing about 0.8%, and the plants, when they are challenged with these elicitors, produce about 2.5% of bixin and not bixin. So what we also saw the expression of some of the genes of the pathway for bixin. I'm not going to the details of this, but what I want to say is these elicitor molecules, when they are applied to the plants, so they increase the production of the metabolites. In this case, the production of bixin was enhanced by 3 folds, and this made the plants which are already cultivated in Andhra Pradesh or in Karnataka becoming a viable source of bixin. Otherwise, India has been importing bixin from South America. But now, with this technology, the standing crops which are presently there in Andhra Pradesh are now able to produce high quantity of bixin. And we have developed a process for downstream processing of bixin and to make as a fine powder. And this process is available from CFTRI. And we have, this is the process details. And it's important to know that this is a permitted color. The toxicity testing has been done. And even the US FDA has given clearance for bixin. And today, this bixin, or the anatro dye, what we call in general, is used to used as a pigment for cheese, butter, or for various soft drinks, and it can also use as a textile dye, and so on and so forth. So, with a simple intervention. Okay, am I clear? Am I audible?
Yes, sir.
Yes.
Okay, so this simple intervention using elicitor, you can increase the metabolite even in the standing crop. So it can be applied to the wide range of medicinal plants to increase the quantity of the drug which it produces. So, and this technology is now used for various different crops, and the this is a very simple and very useful technology. Why is it not moving? Okay, now I would come to the aspect of genetic engineering of plants, which is again the basis for the pathway engineering studies. The genetic engineering or the genetic transformation is done through agrobacterium tumefaciens mediated system. So the agrobacterium tumefaciens has the plasmid, and you can tailor the plasmid by introducing the desired gene and mobilize this into the plant cell. And such a plant cell which is transformed can be regenerated and produce the transgenic plant. So you have the markers, antibiotic markers, or you have different types of markers which are used to select the transformed cell lines and then regenerate that to produce the transgenic plant, and with the desired gene incorporated in this. So this is a very common technique which is adopted, and this has already been used for improving genetic improvement of the plant, for biotic abiotic stresses, for improved nutrition and health in terms of for vitamins, then for proteins and carbohydrates and several others which are produced in transgenic plants to enhance the value of the product. And also it could be for improved plant performance, or it could be for production of certain chemicals, and so on and so forth. And this plant genetic engineering, as you know, has been applied to many food plants, and you have very many vegetable crops, you have cereals, you have pulses. There are many crops which are now genetically modified to have better adaptability for cultivation, and also and the product quality could be improved. So therefore, this is a very versatile tool, and which is already in practice. But well, the permission for extensive cultivation has not happened in various parts of the world for various different reasons. And in India, we have permitted genetically modified cotton for cultivation, and that's for mainly for non-food use. Well, that's a different topic altogether, but I want to say that this is one of the bases for the metabolic engineering studies, that we can engineer the plant in a desired manner. So there are many genetically modified foods like tomato, rice, corn, wheat, and canola, and so on and so forth, which are permitted in some parts of the world, and many countries in Europe and have not approved them. But the issue is being debated, and it is being discussed. So the point is that you can now engineer the plant in a desired manner, and especially when you know the metabolic pathway, you can alter the pathway in the manner we want. And that is the very basis of metabolic engineering. And the strategies used are, once you know the pathway, you can overexpress the step, which is the rate limiting step. So for example, here compound A to B to C is being produced. So you can overexpress the step, which is very crucial for the metabolite production. And you can get higher titers, higher levels of these secondary metabolites. You can also overexpress the genes upstream, that is away at the precursor level, and then produce high quantities of the metabolites. You can also block the undesirable pathway. For example, C is getting converted to D. If you can block the expression of D, you will have higher formation of C. And also you can mobilize this compound C to the storage components. So these are some of the strategies which are used for metabolic engineering. And to do this, this can be achieved by way of regulating rate limiting step, regulation of metabolic flux, expression of multiple genes, targeting the production of desired metabolite through transcriptional factors. So these are some of the strategies which can be adopted for metabolic engineering. Rnai technology is one of the very potent tools to block the undesirable pathway. And here you can make the double-stranded RNA and mobilize this double-stranded RNA into the cell, and then double-stranded RNA gets processed to small interference RNA, siRNAs, and then this together will form RNase-3-like enzyme called dicer, and then this will be leading to RNA-induced silencing complex, RISC, which is produced as a result of the formation of siRNAs and this RISC protein components. And this siRNA strand subsequently guides this domain into complementary mRNA molecules, and where they can destroy and cleave cognate RNA. So these siRNAs will go and bind to these complementary mRNA molecules and result in the cleavage of the cognate mRNA. And this is the RNA interference method, and Rnai technology is used for silencing certain steps, and we have done this effectively with respect to caffeine production, which I'm going to explain to you. And the other technologies which are used is zinc finger technology, and then transcription activator like activator nuclease technology. And in zinc finger, you have the DNA binding domain and restriction endonuclease domains. And similarly, in the talons, you have the DNA binding domains and separate DNA cleavage domains. And these are mobilized for transfer of specific genes, and also to target certain to suppress certain undesirable pathway. So you can also, these are you have, by these technologies, you can have transgenesis, cisgenesis, intragenesis. That is, you can the gene promoter and terminator can be mobilized into the desired host, and this can be in case of transgenesis, the insertion of recombinant element in which one or more components are taken from sexually incompatible gene pools. In case of cisgenesis, it is the gene promoter terminator is from the same gene, and within the sexually compatible gene pool, that means from the same gene, you can take the promoter and you can alter the expression of the desired gene. So you can also use Rnai technology for silencing approach. You can also use and also the regulatory elements and promoters can be regulated for controlled expression. So these are some of the techniques which are used in genetic pathway engineering studies. And these days we have much more powerful tool, that is CRISPR. And CRISPR technology has received a great attention because of its ease in genetic manipulation, and here and then genome editing. And actually this was discovered in the bacteria, and where bacteria, when they were being studied for the from the informatics studies, they found that there were palindromic repeats in the bacterial genome. And this was very intriguing, why these repeats are there. And these repeats are had in between sequences which are unrelated to the bacterium, but later it was found that this was related to the phages, bacteriophages, which are infecting the to the bacterium. And why? And then it was later found that this intervening spacers, they were used as a memory for the bacteria to when the similar type of phage is again attacked, it's used as a memory to block the expression of the genes in the viral vector. So this is an adaptive phenomena which takes place in the bacteria, and which is now used very potently to edit the gene. And so here you have, this is a simple diagram of a CRISPR locus, and which shows the repeats and then the spacers, and this is palindromic repeats and then the spacers, and you have the Cas gene that is responsible for as a Cas proteins for acting as a molecular scissors in genomic in editing the genome. So this is the it was awarded Nobel Prize to these two fantastic scientists, and Charpentier and then Doudna, who received the Nobel Prize, and they showed how parts of the ancient immune system found in bacteria could be repurposed for editing the DNA. So essentially, it has got a guided RNA and the Cas9 complex, and which will recognize the particular locus of the target DNA, and then it causes double-stranded breaks, and then thereby inducing mutation in the gene sequence. This double-stranded break can also be later used to also add on a desired gene into that site, and so therefore you can either add a gene or you can delete a gene from this powerful technology. And so this is also nowadays this are synthesized by various companies. This is from Centenary company, which makes CRISPR cocktail necessary for genome editing. And I'm not going to details of this, but what is important here is that it has got a guide RNA, and it has got the Cas9, and this guide, this and also it has what is called as a PAM sequence, that is a protospace adjacent motif, which is closer to the site where you want to cause where you want to cut this site, and then this guide RNA will go to this site, and then the Cas9 will act as a molecular scissor to cut this. So this technology is now used for genome editing, and which is very powerful, and there is no introduction of a undesirable gene as you see in agrobacterium system. So this CRISPR-Cas9 is a very versatile tool. You can mobilize this into plants through agrobacterium or through protoplasts or through floral dip methods or nanoparticle mediated method or particle bombardment methods. These are some of the techniques which are used to deliver this into the desired plant. So how this pathway engineering has helped in improving the plant for its traits. For example, the cereals are deficient of lysine, pulses are deficient of methionine, and can we enhance this lysine or methionine level in the respective plants? For example, this lysine is always, it exerts a feedback inhibition to the first step from aspartic acid to the aspartate kinase is the step here. And then it also feedback inhibits DPS, that is dihydropicolinate synthase step. And so therefore always this will be limiting the production in the plants. But there are certain bacteria which are insensitive to this feedback inhibition. And if you clone those genes, for example, for aspartate kinase or DPS, it is possible to get plants which are higher in this essential amino acid, lysine or methionine. So you see these examples. This was shown first time in 1995, and the other time it's a very recent study where AK mutants or DPS mutants were constructed and mobilized to rice, and which resulted in lysine biofortification. That means the content of the lysine in rice increased dramatically. That's because it is now insensitive to feedback inhibition. So it gives a beautiful opportunity to enhance the desired metabolites like lysine and methionine. And another very interesting example, which we all know, is the beta-carotene rich rice. And this has been again produced as a result of metabolic engineering with the help of the mobilization of the phytoin synthase, CRT1, and then lycopene cyclase from various different bacteria into rice plant. And beta-carotene levels, then the rice produces beta-carotene, and this will be under the control of tissue-specific promoter. So with this, it has been possible to produce golden rice, that is beta-carotene rich rice. And this is one of the classical examples of the pathway engineering. And we all know that this was recently approved for cultivation in Philippines, and it is going to be a commercial activity to grow golden rice in certain parts of the world. And golden rice also got the approval for safety from US FDA. So because it's not only that functionally it is important, and that it is a source of beta-carotene in a way for vitamin A, and therefore functionally it is useful, but is it also safe? So this question has been addressed, and FDA has approved, saying that this has been found to be safe, and also New Zealand and Canada and many countries have approved for the safety of the golden rice. And now it is International Rice Research Institute director has recently come out with a statement that it meets 30 to 50% of the estimated average requirement of vitamin A. So this is a very important example, and I think even India has a lot to gain from this, but we are not given permission yet for any of the GM foods. So therefore, it is still pending. I would like to give example of from our own study where gamma-linoleic acid can be produced in soybean through metabolic engineering. The GLA is has multiple benefits in treatment or management of asthma, atherosclerosis, cancer, diabetes, depression, for cholesterol management, metabolic syndrome, and several others. So what is important is that GLA has multiple roles, but many of the certain population who are excessively dependent on vegetable sources for food, vegetarian population, they may have a deficiency of GLA. But if they consume fermented foods, the GLA will be met, the requirement of GLA can be met through fermented food. But only from vegetarian sources you can't get GLA. So is there a way to get GLA in vegetable sources? Yes, it is there. For example, GLA production is from the linoleic acid, and most of the oilseed crops produce linoleic acid, but they don't produce GLA. Gamma-linoleic acid is not produced in oilseed crops because this step of conversion of linoleic to gamma-linoleic is lacking in the oilseed crops. And that's because it lacks the gene called delta-6 desaturase. Okay, so this delta-6 desaturase, we have tried to mobilize. We have tried to mobilize the gene for the delta-6 desaturase from a cyanobacterium, spirulina, into soybean. So our lab is working on spirulina in CFTRI, and we have developed the technology for spirulina for industrial production. And so we had these strains with us, and we isolated the gene delta-6 desaturase gene from spirulina and made a construct, agrobacterium construct with agromycin, coliform mosaic virus promoters, with Gus gene and so on. And then we mobilized this into soybean. So upon mobilization and expression of this gene, we could see that the transgenic, and then we grew these transgenic plants in the greenhouse, and we could see that the gamma-linoleic acid was produced in the transformed plants. So GLA was produced at about 3.8% level. 3.8% is the 3.8% of the total fatty acid was GLA. So therefore, it's possible now to produce vegetable oil enriched with GLA. So this is possible through the metabolic engineering, where we borrowed the gene from a cyanobacterium and mobilized into soybean. And what is important is, though we wanted GLA to be expressed, and that is desirable, is there any unintended effects of this genetic engineering or the pathway engineering? So one of the studies we did was to see whether the isoflavones which are present in soybean, is it altered? So we could see that the isoflavone, this is the control and this is the transformed one, the isoflavone levels did not become lower. So therefore, this very important nutraceutical isoflavones are also produced in these transgenic plants. So this is another example, right, from our study where the metabolic engineering could be used for value addition. And we have also worked on another system, that is production of caffeine and regulation of caffeine, I mean, through pathway engineering. See, decaffeination is a process which is adapted by the food industry for removal of caffeine from coffee. And in fact, some people are sensitive to caffeine, and therefore such individuals need to have decaffeinated coffee. So for this, we have a decaffeination process, which is essentially based on solvent extraction method. And however, you have the problem of the residual solvent there. So if you can metabolically engineer the pathway, you can have the caffeine lowered or caffeine free coffee without any such disadvantage of having residual solvent. So for this purpose, what we did was we studied we had the understanding of the pathway, and it is from xanthocene to caffeine. There are 3 methyl transferases, xanthocene to 7-methylxanthine, xanthone, xanthine, and then theobromine, and then caffeine. So you have 1 methyl transferase here, 1 methyl transferase after theobromine, and upstream you have from xanthocene to 7-methylxanthine. So what we did was in this project, which was a multi-institutional project, where we collaborated with a number of institutions like University of Agricultural Sciences, Bangalore, Madurai Kamaraj University, and Coffee Board, and several others. And we were we took the mandate to work on the caffeine biosciences, but other people worked on different other things like the adaptation of the plant and so on and so forth, enhanced root production, or through certain genetic engineering studies, or through different applications of coffee they were looking at. But we were interested in the regulation of caffeine. So and we have published a number of papers in this area. And what is important is that we developed somatic embryogenesis system in coffee. And this somatic embryogenesis system is so interesting. You don't need to wait for the plant to produce caffeine after setting the fruits or after the seed setting. So somatic embryo itself will produce caffeine. So if you have a process to interfere with this caffeine pathway, it will show up in the somatic embryos. So therefore, you have a model system here, and which is also a wonderful system to understand the regulation of caffeine. So what we did was we had 3 types of constructs. One is for the methyl transferase, and where we had one in the sense construct, another is antisense construct, another is a RNAi construct, that is inverted repeat construct. So we mobilized these, we made these 3 constructs in the agrobacterium, mobilized this to coffee, and we developed the somatic embryos, and we could see that this is the normal somatic embryo, and this is the invert repeat mobilized somatic embryo. This is an antisense construct mobilized somatic embryos. So you can see that the caffeine level here is lower when compared to control. So this is the control. These are the transformed ones. So we have now, using this methodology, to lower the caffeine in the coffee plants through RNAi technique or the antisense RNA approach. So therefore, we have now able to produce these transgenic plants, and also we have given this transgenic plant to Coffee Board, and they are now presently growing them in the greenhouses, and studies are going on. And we at CFTRI had no permission to grow them beyond a laboratory level. And the coffee board has the permission from GEAC for the study. So this is another example where you can use the metabolic engineering to alter the pathway. And in this case, metabolite, which is somewhat undesirable, or rather undesirable for a particular product, we have targeted that. In this case, lowering of caffeine, we have targeted. So we have also worked on the promoter of this theobromine synthase, and we have published this work. And then studies are going on in CFTRI now, promoter-mediated silencing of the pathway and so on and so forth. I'm not going into the details of that, but there are innumerable possibilities that way. So from here, I come to the algal studies, which we have been doing, and where alga is used for various different purposes. For example, spirulina for health foods, dunalilla for as a source of beta-carotene, Hematococcus for astaxanthin. In fact, we have recently come up with algal technology volumes for CRC press. It has come in 2 volumes. Volume 1 on food health and nutraceuticals, volume 2 on phytoremediation, biofuels, and biomass production, and so on. So algae are useful for health foods and also for specialty chemicals. And some of the algal forms are also grown commercially to produce DHA, docosahexaenoic acid and pentanoic acid, which are essential fatty acids required for, especially DHA is needed for the growth of infants and so on. So therefore, infant food formulation using the algal DHA is commercially now available. So we have worked on spirulina technology, and we have worked on large-scale production of this spirulina technology. We have also commercialized this technology to industry, and from our efforts, Asia's largest production of spirulina was done by the Topper group of companies, and later several other companies like Parry company has worked on this, and they are the leaders today in production of spirulina, dunalilla, and Hematococcus. So this spirulina can also be used for the pigment phycocyanin. Just trying to give some of the applications of this cultivation of spirulina. So this is phycocyanin extracted from spirulina, which can be used for food and also cosmetic applications. So we have some publications from this, and also we have patented this technologies. And this is used for, as I said, for cosmetics and also for food applications. And it also has anti-tumor and immunomodulating activities. And so mainly this is used as food colorant. And this has also been permitted color by FDA, and FDA has given permission. And therefore it's used presently in very many food formulations. And so this also the FDA approval. And we received a number of awards in recognition of our studies on algae and plant cell and tissue culture for value addition. These are some of these. And we have also developed technology for dunalilla. Dunalilla is a green alga which produces beta-carotene. And there is another alga called Hematococcus, a green alga which produces astaxanthin. So this dunalilla is a halotolerant form of green alga. It can grow even on very high salt concentrations. It can even the salt basins. In salt basins, you can see the formation of growth of this alga dunalilla. So whereas Hematococcus is green, is a freshwater form, and it produces another very interesting molecule called astaxanthin. And so beta-carotene to astaxanthin is a 2-step process. you have a beta-carotene ketolase and then hydroxylase, which produces astaxanthin. And we have a process for the production of beta-carotene here. And these are commercial production of beta-carotene and astaxanthin from it's the Paris nutraceutical company, which is growing this and producing in large scale. And they have really high quality beta-carotene and astaxanthin. And today you have many formulations internationally. And astaxanthin is very potent antioxidant and also white, it can, it's also said that it can cross the blood-brain barrier, and it is much more potent than vitamin C or beta-carotene, and because it can, because of the structural advantage it has over beta-carotene and vitamin C. So this is astaxanthin is also useful as a photoprotectant, with anti-cancer properties, anti-inflammatory properties, and so on. So we have done the downstream processing of this, of the astaxanthin, and we have also found that this astaxanthin has got anti-tumor effects, and here it is the tumor-induced skin tumor-induced, and it's healing through application of astaxanthin. So I'm not going to details of this, but these studies we have already done in our lab, and we have also published these studies we have reviewed this in marine drugs, and this particular paper in marine drugs on astaxanthin has received 26,000 downloads as of today, and it has received about 800 citations so far. So that shows the importance of this molecule and the importance of the technology for commercial use. And we also came up with a book on astaxanthin, and very recently it was published in April this year, and by the academic press. So what is important with respect to the metabolic engineering here is that the two beta-carotene and beta-carotene hydroxylase and beta-carotene ketolase, which are important for making astaxanthin, can be mobilized from Haematococcus to Dunaliella. Dunaliella, I told you, produces very high quantity of beta-carotene, but it doesn't produce astaxanthin. So what we did was we isolated the gene from Haematococcus and mobilized into Dunaliella. So I'll give you the next few, and we have also done the transformation studies to agrobacterium mediated transformation of Haematococcus, and we have also published this study on the expression of astaxanthin gene in this Journal of Biotechnology. And what I want to say is beta-carotene ketolase, when it is mobilized from Haematococcus into Dunaliella, and so this is the construct we have made, and the transformed Dunaliella start making the astaxanthin. So the expression of astaxanthin in Dunaliella we have shown, and this provides an opportunity to produce astaxanthin in a newer host, that is Dunaliella. So it's an interesting study, and it is still a long way to go to make it commercialized. But what is important is that this is now feasible, that is mobilizing beta-carotene ketolase into Dunaliella to produce the astaxanthin in a newer host. Okay, so, and such metabolically engineered organism, they cannot be grown outdoor. They should be grown in contained environment. So they can be grown in bioreactors like this, but we do not have the technology for this in India. And therefore, I think it's necessary that we develop technologies for production of these algal forms, microalgal forms in large quantities through photobioreactor methods. So now you have seen golden rice being produces beta-carotene. I have shown now example where mobilizing the beta-carotene ketolase, beta-carotene hydroxylase could produce astaxanthin in a newer host. So these constructs could now be used even to produce astaxanthin in rice. So therefore, that's because the precursor beta-carotene, which is required in rice, is already now produced in golden rice. So if you transform this golden rice with the inclusion of beta-carotene ketolase or beta-carotene hydroxylase, you can produce even astaxanthin in rice. So this could be a possibility in future. So again, I'll take another 10 minutes. Is it okay? Hello?
Yes, sir.
Yes,
I'll
it
take
is
another
okay.
10,
Yeah,
10 or
Yes, you
15
can.
minutes.
fine.
Yeah, please tell me. Okay, is that okay?
Yes, sir. You can.
Okay.
Absolutely okay.
Okay. Okay. So this is another study which we of the capsaicin pathway, which we have done in our lab. And what is important is capsaicin pathway has two branches. One is the phenyl propanide pathway. Another is fatty acid pathway. The phenyl propanide pathway from phenylalanine all the way to vanillamine, and vanillamine combines with 8-methyl nanoic acid to produce capsaicin. So what we have done in our lab is to identify the gene responsible for vanillin to vanillamine, and in order to regulate capsaicin production. But I would tell you that this study has a newer angle to produce novel molecules. Instead of capsaicin, you can produce capsinoids. So I will explain that in the next couple of slides. So before that, I will just briefly mention about our studies on vanillamine, this step, vanillin to vanillamine step. This is a methyl transferase, so amino transferase, sorry. Vanillamine amino transferase producing vanillamine. So we have purified the vanillamine transferase enzyme and done the N-terminal sequencing of the amino acids. And then we have also, this is the sequence of the putative amino transferase. And then we have also isolated the gene for vanillamine transferase from the capsicum plants. And we have also seen the expression of this putative, this amino transferase in the fruits of capsicum plants, and which coincides. This is the 30 days after anthesis. This you will see that the maximum quantity of this gene is there, and also this coincides with the formation of capsaicin. So therefore, we are able to identify a very important gene in the pathway. And what is important from the point of view of metabolic engineering is, for example, instead of vanillamine here, if you had vanillyl alcohol, so you get this with the fatty acid, you will get a novel molecule called capsinoids. I'll show you that. So these are capsinoid molecules which can be produced from a different substrate. Instead of vanillamine, you should have vanillyl alcohol. So what is importance of the capsinoid? Capsinoid is an anti-obesity molecule. This is in high demand. It doesn't have the pungency as capsaicin has, and it can be used in obesity management. This has been already patented by the Japanese, and I was told that they are commercially producing this through the metabolic engineering. So you see here one publication again from Japan, an enzymatic synthesis of capsaicin by acylation of vanillyl alcohol with fatty acids. So therefore, this kind of possibility opens up when we have the understanding of the pathway and when we have the understanding to tinker the pathway in a desired manner. So this is another wonderful application which I see of metabolic engineering, which can produce novel molecules, capsaicinoids in newer hosts. You can produce this, and also you can use, once you produce this, you can even see whether you can make it through biotransformation and so on and so forth, but you can produce in situ in the plant itself, so it can be widely applied. And extending this study, you can also make tomato to produce capsaicin. So that is, even tomato has all these pathway in many of these steps, I mean, not all, many of the steps. But if you can express the pathway like the phenylpropanide pathway, and also mobilize this final step condensation pathway of condensing the fatty acid and the phenylpropanide compound, that is vanillamine, you can produce the capsaicin. So therefore, you can make, just as chili, you can make even tomato produce capsaicin, or you can alter it in such a way that it can produce capsaicinoids. So there are wonderful opportunities here to produce novel compounds. So this is another futuristic example, but this paper came in 2019, Trends in Plant Sciences, and this was a, they have predicted, it's not from our group, it is from some other group, so in the US. So they have predicted that hot tomatoes can be produced through metabolic engineering of the capsaicin pathway in tomato. Okay, so then heterologous expression is another important area. You can produce recombinant proteins in a newer host, and there are many examples of that. These are some of the metabolically engineered plant products, which are again in different levels of production. It could be botriococcines, these are bioenergy molecules, betalins, resveratrol, and so on and so forth. So this has been summarized in current opinions in biotechnology. And so therefore, there are innumerable opportunities for the future. So these are some of the products from the heterologous hosts. For example, insulin, alpha-1 antitrypsin, interferon, and so on and so forth. So all these can be produced in newer hosts and for various applications, for medicinal, for industrial use, or for food application, and so on and so forth. Finally, another 5 minutes I'll take for this another important topic, that of synthetic biology approach to produce phytochemicals. And this is with respect to artemisinin production. This lady, Tu Yu, China, received the Nobel Prize for artemisinin in 2015. She received the Nobel Prize. She says she found the artemisinin reference to the importance of anti-malarial compound in Artemisia through the Chinese text. So she says, when reading through the Chinese text, she came to know of the importance of Artemisia for malaria, malaria treatment. And this discovery, and later the studies on the efficacy of artemisinin as an anti-malarial drug earned her a Nobel Prize. So since it can be produced only in Artemisia plant, and a synthetic biology approach to produce artemisinin in bioreactor has been planned, and it was funded by Bill and Melinda Gates Foundation. So what is important here is the Artemisinin pathway goes through the formation of amorphodiene, and amorphodiene gets converted to artemisinic acid. So this pathway can now be expressed in yeast. So what has been done is the parent yeast strain has been mobilized with the genes for amorphodiene production from Artemisia, and which produces high titers of amorphodiene. It produces up to 40 grams per liter, which was unthinkable for production in the plants. So on this, after amorphodiene production, the genes for hydrocarbon oxidation was introduced, and then further this improved the artemisinic acid production, and further addition of Artemisia annua gene for oxidation was again introduced, and this leading to very high production of artemisinic acid, 25 grams per liter. So the precursor 40 grams per liter, and here the product is 25 grams per liter. And this is later chemically processed to produce artemisinin. This is again another success story through this kind of semi-synthetic route involving the approach to the synthetic biology for production of phytochemicals. So in conclusion, I would like to say that metabolic engineering has many applications. It could be in food, it could be in medicinals, it could be in various different compounds which are of use for industrial use, and novel compounds which were not produced in the original plant can be produced, or it can be even produced in a newer host. So novel plant-derived drugs or novel production technologies will surely see an upsurge in research interest, and the scale-up methodologies and sustainable production will continue to receive much needed impetus, so that because these molecules, these organisms rather, which produce novel molecules cannot be grown in open environment. They have to be in closed environment. So if we can provide use cell culture methodologies to produce this in contained vessels, so you can scale it up and exploit this for production of metabolites. And genome editing will become increasingly adapted. I showed you the genome editing technology. Still, there is a lot of opposition for this, but slowly the acceptance is gaining. I would say that one day genome editing will become very common tool to improve the plant or improve the organisms, or even help in for human diseases and so on. So therefore, this will be a very important tool. And then synthetic biology approach will again become very important, and you will see a number of more products from this. And most importantly, in all these studies, the safety. So safety evaluation is absolutely necessary. You've seen in golden rice that safety evaluation was thoroughly done, and it has been shown to be safe from independent investigations, and therefore now it's accepted by the Philippines to grow the golden rice. And similarly, in case of transgenic plants, for example, BT brinjal has been now accepted in Bangladesh for cultivation. So it's slowly many countries are clearing the regulatory hurdles after thorough examination of safety. And so therefore safety is fundamental to apply this technology widely and benefit from this technology. So with this, I want to conclude my talk. And before that, it's my duty to thank all my colleagues in Plant Cell Biotechnology Department, CFTRI, and a number of PhD students who are involved in these studies. In fact, what I presented to you is from more than 20 PhD theses, which has emerged from our lab. And so I'm grateful to all of them and also a number of funding agencies. And I also thank once again the organizers of this symposium for giving me this opportunity to present the perspectives of metabolic engineering, and which will have innumerable opportunities for future. And I'm sure many youngsters who are watching this will see, will take interest in this, and surely will be benefiting by applying with curiosity the knowledge of the metabolic pathway to be tailored to make novel compounds, and this will be a big value addition, and which will be of use to the industry and for economic growth. So with this, again, I thank everyone who has been patiently listening to my talk, and I also thank the organizers once again. Thank you very much. And if you have any questions, I'll be happy to answer, or if you have clarifications, I would like to clarify the same. Thank you very much. Yeah, thanks.
Thank you very much for sharing your profound perspectives with us, sir. We are sure that this session would continue to remain very helpful and applicable for us in the time to come. Just a reminder, please be sure to type your questions into the live chat box. We already have a few questions. Sir, let us now move on to the question answer session.
Yeah, please.
So there's a question from Manoj Kumar. Can secondary metabolites be used for having canola quality in mustard by alternative fatty acid pathways?
Oh, again, for, can I, sorry, I couldn't.
Can secondary metabolites be used for having canola quality in mustard by
canola?
Yes, sir.
Oh, yeah, it is possible. See, you have to, firstly, we have to understand the pathway, number one. We should know what kind of trait, what kind of quality you want to introduce, or you want to remove some undesirable metabolite from that. You need to know what you are tinkering. So once you know what you are tinkering, you can devise a method accordingly for that. So I don't see any reason why we should not use the metabolic pathway engineering to do that.
Okay, thank you, sir. He has got one more question. Is there any secondary metabolites for curing coma patients suffering from axonal diffused injury? Is spirulina have any role in healing brain injury?
Well, it's this is actually there are not many studies on this, and so I cannot really competently answer that question. So spirulina is used for very many things, like as a immune booster, as a source of nutraceuticals, or nutritionally important compounds, and so on and so forth. But when it comes to clinical applications, you need to have very in-depth studies. So therefore, today, spirulina is used as a health food. It's not used for any drug application, in my opinion. Yeah.
Thank you, sir. Another question from Jana Navede. What kind of microbial elicitors we can use?
Yeah, okay, good question. See, microbial elicitor could be from as I said, it need not be pathogenic. So the best is to use a non-pathogenic one. We used the Rhizopus, Oligosporus, we used Aspergillus, which was not pathogenic. So we used things like this. Even people have used microalgae also as agent for elicitation. So essentially, it's the cell wall polysaccharides which are in which are the inductive factors, so inducive factors. So therefore, it need not be live. Also, you can make extracts from the cell walls and can use as elicitors. So we have used extensively the Aspergillus and Rhizopus in various different systems. I showed you about the Bixa system, but we have also used that for many others, like soybean to enhance isoflavone production. So similarly in few medicinal plants. Yeah. So you can use different ones, but if what you can do is try it out in a cell culture system. It's easy to try that and see whether your metabolite yield is higher. Once your metabolite yield is higher with certain fraction of the elicitor, you can always apply this in field conditions to increase the metabolite content of the field grown plant.
Okay, thank you, sir. There is a question from Dr. Sudhamai Behura. Can we think for the use of secondary metabolites to get rid from this corona pandemic?
Okay, so actually several studies have been done these days, and there were many studies using the bioinformatics, and also some studies, involving docking studies for which will specifically target to the virus, viral protein, and so on and so forth. So there are many reports, but none of them really could be used as a therapeutic agent. But what was important was some of the plant-derived the formulations which they made, they possibly helped in enhancing the immunity. So spirulina was also projected for that. CFTRA did, there was some study where spirulina increases the immunity. So they made spirulina chickies and so on and so forth. And then it was marketed. So what I want to say is, for immune boosting principles, it has been tried, but not directly as a drug for corona.
Thank you, sir. There is a question from Dr. Chinju Bose. She's telling thank you for sharing your vast experience, sir. Castor shell oil is a huge bioresource that is available in Kerala, and she had developed equipped methods for isolating ancardic acid, cardol, cadenol from this. So do you see any commercial importance for these compounds from your experience?
Yeah, see, there are innumerable possibilities of such studies of medicinal plants of which have been consumed, which have been used for various ailments. So you can extract the metabolites, but all you need to do is validate the bioefficacy, validate the administration method, and so on and so forth. And once you have a very potent plant-derived molecule, you can always commercialize that. I don't see any reason why we should not be looking at that. And in fact, the Chinese are coming up very aggressively, coming up with several such herbal medicines, and they are marketing worldwide. Yeah.
Thank you, sir. There's another question from Sudhi Singh. What is the status of acceptance of genetically engineered enhanced secondary metabolites production, especially if they are grown in contained or bioreactor condition across globe or India, assuming efficacy of the product of industry is not an issue?
Yes, it is. So far, the genetically, the plant drugs derived from genetically engineered plants has not been, they're not taken it up to that level. I think that the thing is that it is like once you get a molecule from GM plant or non-GM plant, it's the same. It's the same. The only thing is, how do you grow these plants, whether you grow them in contained environment, or you have any issues of from the environmental point of view, or do you have have you really tested the toxicity and so on and so forth? Even though you may be dealing with the same molecule, you need to really work on all these things. For example, we know that beta-carotene is safe, but they had to show that beta-carotene in rice was also safe. So therefore, the safety studies were done. So therefore, safety studies have to be done, and then it needs to be put forth accordingly. I'm sure there is not far off when we'll be using many of these for applications.
Thank you, sir. Any more questions? Sir, it looks like we have covered all of our questions. Is there anything else you would like to mention, sir?
No, myself, no. I'm very thankful to you for this opportunity. That's it. Especially Susan, she did the networking and in fact, I changed the timings and so on and so forth a couple of times, and she accepted all that. And therefore, it was possible to have this lecture today. So thank you, Susan.
Sir.
And thanks, Vijayan sir is sitting through. He's such an inspiring person. And when he is there, I know that the session would go very well. So
yeah,
and,
it was nice.
Thank you very much.
Nice of you to have
thank you very much.
Given us such a nice presentation.
Once again, thank you, sir. We are so glad to have you today, sir. I would now like to invite Dr. Deepu Matthew, Secretary, NACI Kerala Chapter.
Thank you so much. Truly, today has been a great day to hear from a great scientist. So, thank you so much for detailing on the exciting works from your lab, like the production of transgenics with low caffeine content, then algal technologies, similarly astaxanthin, then the emerging field of capsaicinoids. So, sir, we have really enjoyed the work as well as your talk. And above all, I should sincerely thank you for accepting our invitation and taking so much of time for preparing and giving such a nice talk. Thank you so much, sir.
Thank you. Thank you.
Apart from that, I should thank the manager of UC College. So we know Reverend Thomas John, who has been giving continuous support for the bioscience team as well as the faculty in the UC College, and we thank him. And also the principal of UC College, Dr. Tara Simon, and she has been arranging all the things for this sort of a great talk. And I'm not sure whether I should thank Dr. Susan Eapen, because she has been everything for this talk. Maybe from the past 2 or 3 months, she has been talking to us, arranging each and everything. Even yesterday late night and today morning, I had messages, continuous messages, managing each and everything. So I think that is the real support a great scientist can offer for this sort of a program. So, Madam, thank you so much for the care you have been taking. And now I should thank the head of the Department of Bioscience in UC College, Syam Mohan. And also I should thank the organizing secretary, Dr. Sareen, and also Ms. Ashwati, who has been comparing this program very beautifully. Thank you, Ashwati. And as you know, Nasi Pragyaraj is committed to do its societal dedications. And especially, I was really thrilled to see a Nasi fellow is giving the talk. Nasi fellow is organizing the talk, and Nasi is sponsoring the talk. So that's the level we have reached. And from everywhere we are getting lot of support. And I should thank Professor K.V. Peter. And since he has been traveling today, he couldn't give a nice talk today. And he is actually guiding the Nasi Kerala chapter. And my sincere thanks I am placing in front of him. And also our coordinator, Professor Vijayan sir, is here. And sir is actually guiding every program. So for every program, I'll be calling him, and he will be so busy. He'll be managing many programs. Even after all this, he will take time to attend and see his presence, give the valuable guidance. And I do not know actually how to thank Vijayan sir. Oh. And again, yeah, and again. We have the advisor for the chapter, Dr. E.V. Sonia from Rajiv Gandhi Center for Biotechnology, and all the fellows and members of NASI. Thank you all for the support you have been giving. And I must thank all the participants. You are the actual strength. And the question and answer session, it has actually shown to what extent or the great care the students as well as the faculty and all the participants were giving to the talk. So that this was a great event, and I am thanking every faculty in the UC College and all my colleagues from Kerala Agricultural University who all have joined this program and made this a great event. Thank you so much.
Thank you, sir. Thank you for participating in today's webinar. We hope you have learned and enjoyed this presentation. Now, a Google Forms link will be available in the live chat box, and we would appreciate it if you can complete that and provide your feedback. After that, you will receive the email within 24 to 48 hours with an e-certificate. The link will be active only for 15 minutes. An announcement is that there will be a seminar on 9th December at 10:30 a.m. by Anjan Banerjee, FNASC, FASC D, ISA Pune, sponsored by NASI. We have now come to the end of this session. I wish you all a time full of positivity and productivity. Thank you once again.
It's on 9th October.
Thank you, sir. Thank you.
Thank you
so much.
Thank you, Susan.
Thank you, Susan. For you to sleep, I suppose.
Thank you very much. Yeah.
Good night.
1:30, 2 o'clock.
Yeah, 2 o'clock.